So I saw your general comments about the podcasts, and most of you, pretty much all of you, got full credit for that portion of the quiz. But I have here a couple notes that I made. Were you skeptical of anything in those podcasts? Did any of them say? So many of you were persuaded by the information that was given to you, liked the presentation style to a greater or lesser degree. Nobody gave anything lower than a B of some sort. Maybe there was someone who gave like a 75 to one of them, but no one gave lower than that. The reason I'm asking this is because I was skeptical of a couple things in there. Hey, Damon, just walk through it. It's okay. So for example, there was the example where he was talking about a super marathon runner who was vegetarian and who had been doing it for years. And they indicated that guy had a huge protein requirement. And even this guy, who had an enormous protein requirement, was able to eat enough protein out of their diet. Right? What did I say about nitrogen balance? Think about the facts that you know that I gave you in class. Does anything about that seem dubious to you? Was this super marathon runner, what does it take to be in, first of all, what is a positive nitrogen balance? Yeah, when you're building muscle mass, right? And so what are some examples of that I gave you? I gave you 3 in class. Bodybuilder or athlete of some sort that's in training, building muscle mass. What's another one? Kids and pregnant women. Those were 3 examples of positive nitrogen balance. Is a super marathon runner who's been doing it for decades putting on muscle mass? Not at all. Is that person likely to be in positive nitrogen balance? Not at all. Does that person need an enormous amount of protein? Not at all. Not at all. So there were several other things like that in that podcast where those people were speaking convincingly, were speaking in a very casual style that put the listener at ease. More of you rated the second podcast higher. I didn't take the time to actually record the grades that you gave them, but just noticing more of you gave that second podcast a higher score than the first podcast. And there were several instances of blatantly factually incorrect information that those guys were putting into the podcast, essentially spreading misinformation. What was their agenda? About half of you got it. And half of you, when I say agenda, who are they speaking to? I'm kind of making it political when I say agenda. Yeah. Yeah.
Also, athletes, there has been people who are pretty much them. And so I think it's also interesting, they probably want to confirm their lifestyle choices.
Right.
They're kind of doing research to support those choices and also kind of putting it as they definitely made it seem like it was the best lifestyle.
Yeah, confirmation bias is what we call that. Yeah, it was they were looking for facts and interpreting facts, and then also saying unfactitious, factual things, fictitious things, to support their perspective. This isn't to say that there aren't a lot of things to recommend a vegetarian diet, and that a vegetarian can't be an elite athlete, because certainly they can. But they were trying to create a narrative that was convincing to the audience. And so one of the reasons I put that podcast in there was to see if I could trip any of you guys up. They also talked about the fact that no one in America is at risk of protein deficiency, having a varied diet. And Americans have such a varied diet that they're unlikely to have a protein deficiency. How does that reconcile with some of the data that I've shown you in our nitrogen unit? Yeah.
Seems like a pretty broad substance. I would say, I've never seen it one of the possibilities. I've never seen it in just one area.
Yeah. Where did they live? Did you pick that up?
I think I remember now.
They lived in Boulder. Oh yeah, Boulder or Denver, right? I forget. I thought it was Boulder. Yeah, in a wealthy urban area in Colorado.
Seems like the whole country, just because you haven't necessarily seen it in your small Denver community, doesn't mean anywhere else in the United States that's not.
Absolutely, there are people in this country who are malnourished. There are people in this country who are protein deficient, and just because those guys live in a sequestered elite environment there doesn't mean that their experience is necessarily indicative of broad policy recommendations that the government, the U.S. government, should take, for example. So the other podcast didn't have as much detail, did it? It didn't have as much detail, in-depth science and facts. A lot of you guys really liked those facts, which makes me feel good that you are being persuaded by data. But part of the point of me putting those two podcasts there, one which was a little bit light on hardcore scientific facts, but they didn't say anything untrue. And they did stimulate an interest in the topic, and then provided more resources if you really were interested on their website where you could hunt some stuff down, versus another where they threw a bunch of facts out there, many of which were true and some of which were not, to let you know that it's not just facts. Science is not based just on facts. Having a body of facts is not science. Being able to skeptically analyze, critically think about facts. So that was the first point. That's part of science. And then the second point is the responsibility of the journalist, the person telling the story, to try to make sure that your mind is open, that you've looked at different sides of the story, and in your zeal to tell a certain story, that you don't fall into a trap, that you don't fall into the trap of misinforming or letting your own misunderstanding of a topic perpetuate itself. Those guys in that podcast were talking about how they had just read a lot of this material that morning and were talking about it. Does that necessarily speak to a level of in-depth understanding of the material? Not in my mind, necessarily. So it wasn't a terrible podcast. I wouldn't have given you a terrible podcast, but it had just enough red flags in my mind when I listened to that one. I was like, oh, okay. I want to put this out there so that you are aware of that when you're making your own podcasts. All right? Be aware of the story you're putting together, the facts that you're collecting. Because it's hard, particularly in nutrition science. We're still trying, the scientific community, the nutritional community as a whole, is still highly conflicted over a lot of issues. And that's partially because we don't fully understand a lot of the biology yet. We're still grappling to understand the biology. Oftentimes, biology is individual dependent. So, all right. Yeah, I had a bunch of other points there. The one about the being the impossible to be nutritionally insufficient in America. That stuck in my craw just because I grew up in a community where that was not true. So, yeah, reconciling a lot of this with your own experience is important as well. All right, I'm going to pick back up here where we left off last time. And I'm going to talk through the end of proteins. I decided to take some of the material that I cut out from last week and put it back in just a few slides, 5 of them, which will be on the test. And then when I start carbs at the end of the hour, or if I don't even get to it, it's no problem. But none of the carbohydrates will be on the test on Friday. I'm going to push that into the next section. So just up to proteins will be on the test for Friday. Okay? There had been 2 podcasts for Friday. I cut it back to 1 podcast because of that. And there will be a question, or probably 1 question from the podcast on the test as well. So it'll be sort of a combined podcast quiz and test. But just on the podcast for this week, it's short. It's only a half hour. It's an interesting story, and it's not really heavy in scientific details. There's some in there, but not an enormous amount. It's about proteins. Yeah, Peg. Nope, I've already quizzed you on those. Unless the material in the podcast is something that I covered in lecture, and if it's been in lecture, I've talked about it, it's fair game. All right. So let's interrogate this idea of combining complementary proteins a little bit and see what the deal is that these guys were talking about. So who originally came up with this idea of combining complementary proteins? Well, there was an article in 1952, a scientific article by a nutritionist named William Rose that recorded a minimum daily requirement of 8 essential amino acids. These are amino acids that mammals do not produce. We saw them already. And then he estimated how much you need as a basic steady state, and then he just put them as daily requirements. He took those minimum amounts, he doubled them, and then said that's what he recommended, a recommended daily requirement in 1952. If that's too loud for you, you can unplug it or whatever. Is that okay? Oh, push the screen around if you need to. If you need to tilt it a little bit. More of you are on that side than over here. Yeah, they can move if they don't like it. All right. So then in 1971, this smiling woman here, named Frances Moore Lepay, published this incredibly influential book, Diet for a Small Planet. And I was a vegetarian I've been a vegetarian. I'm not currently, but for almost half my life now, I was a vegetarian. And essentially because of this book. I read this book when I was even younger than you guys. I was 17. I read the book, said, hell yeah, okay, I agree, I'm in. And her general premise was that there's this exploding population, and we put an enormous amount of resources into growing meat, and producing meat is an intrinsically inefficient process. She claimed it was unsustainable, rightly so, unsustainable for the long term, and said that humans can get all of the protein they need. They can get all of the nutrients they need by being vegetarian. In this book, that was the central thesis of this book, central thesis of her doctoral dissertation. She was doing graduate work, and that dissertation expanded into this wildly successful book in 1971. In it, she talked about food combining, picking up in her research on this article by William Rose from 1952, 19 years earlier. And she talked about combining grains with seeds, or grains with legumes, or seeds with legume, like doing different ways of of combining different food groups within a meal to get all the essential amino acids that you need in a day, in a meal. She believed in, she advocated this complementary protein thing. So that's a scan from the book there, a page torn from the book on the right-hand side. 4 years later, Vogue magazine decided to make it Vogue. And so here we have on the cover of February 1975, The Secret of Meatless Eating So You Feel Better, Spend Less Money. And they picked up on this idea of food combining complementary proteins. It was then reinforced, picked up out of Vogue, and then reinforced in the medical community by an article that happened several months later in December of 1975 in the American Journal of Nursing. There's this article, Making Vegetarian Diets Nutritious. Their diets can be adequate, palatable, and appealing to the eye, but vegetarians need considerable information to choose foods that provide the essential amino acids, vitamin B12, and other nutrients. By Eleanor Williams, thus kind of canonizing the notion that each meal needs to have the complete complement of essential amino acids. It turns out that this is not actually the case, that those speakers in the second podcast were actually correct, that the typical American diet has enough variety in it if you are a vegetarian, as long as you're not eating nasty food, that you are going to, within the broader arc of time, across multiple days, you're going to get enough of the various essential amino acids that you need. If you don't eat enough lysine in a day, chances are you'll get more than enough tomorrow. All right? And that's the basic concept behind what they were saying in the podcast. In the follow-up to this book in 1981, here's Frances Moore Lepay 10 years later, in her early 80s turtleneck, saying, in 71, I stressed protein complementarity because I assumed that the only way to get enough protein was to create a protein as usable by the body as animal protein. Combating that myth, that meat is the only way to get high-quality protein, I reinforced another myth. I gave the impression that in order to get enough protein without meat, considerable care was needed in choosing food. Actually, it's much easier than I thought, with 3 important exceptions. There's little danger of protein deficiency in a plant food diet. Those exceptions are diets very heavily dependent on 1, fruit, if you're just eating fruit, 2, some tubers such as sweet potatoes or cassava, which are dramatically deficient, and so this is going to be in indigenous cultures that have a very narrow diet, or in the American version of the indigenous diet, the junk food, refined flour, sugars, and fat, basically garbage. So being a vegetarian doesn't mean you should eat garbage. Fortunately, relatively few people in the world try to survive on diets, etc., etc. So now, people still have not fully embraced this idea that food complementarity is not something that needs to be on a high granularity of daily combinations. Although this is not to say that you do not need to have complementary sources of protein across longer time scales. You do. Essential amino acids are still essential. You need to get enough of them. But getting enough of one or another at a single meal or in a single day or even a single week are probably not essential, not critical. Yeah?
I think about the fruit. I know in the podcast they definitely talked about like a lot of fruit, like people like raw food vegan.
Yeah.
And so I was like, I have heard doing that, and I was wondering, like, is that what's your opinion on that? Is that mean that like strictly like medical work, or like work to like take more time?
Your question is too general to give a specific answer to, because it's going to depend on, number one, the fruits you're talking about. Secondly, the nutritional needs of the person. Is the person in nitrogen
surplus or deficit? Are they in a positive nitrogen balance or negative nitrogen balance? Are they putting on muscle mass? If they are putting on muscle mass, what kind of fruits are they eating? What is their body weight? What kind of fruits are they eating exclusively? And does that fruit have enough of whatever is sent, is it a broad enough profile of fruits that you're able to get enough protein to satisfy your nutritional needs? When she's saying fruit, she's talking about communities that are on subsistence growing of agriculture and fruits that are right around them, right? And are they getting a varied enough diet? So maybe this was some sort of community that at one point were hunter-gatherers, and the game is not there. They're not able to access animal sources of protein now, and they're relying pretty heavily on maybe some kind of monoculture of fruit that's been established because of, I don't know, who knows, some sort of colonial corporate agriculture that has come into their, I don't know. Fruit is not particularly high in protein. And so you are at higher risk of protein deficiency if you're eating exclusively fruit. That's my. Does that answer your question?
I was just wondering, like, in the podcast, like, if that was the things that were not really factual about what's making it seem like you could just have, like, a raw food diet, like, buy a lot of protein, like, you can get protein.
Yeah, well, and one of the other things about it is protein absorption in raw foods. I mean, I've had a raw food diet. They're interesting. There's some interesting things about raw food diets, but raw food diet, the absorption of amino acids is lower in raw foods than it is in a food that's been partially denatured already. So, yeah, I mean, you do get other benefits from eating raw foods, for sure. I'm not telling you that everything should be cooked. In fact, everything should not be cooked that you eat. But if you are eating exclusively a raw fruit diet, it seems like that would be a scenario that could put you at jeopardy of a little bit of protein deficiency, particularly if you are in a positive nitrogen balance for some reason. All right? Any other questions? It's interesting. It's interesting to think about, because this was one of the canons of vegetarianism for a long time. I know there's a couple of vegetarians in the classroom. I was one for many years, but in the context that I became a vegetarian is different than the current thinking. So it has changed. This woman, Frances Moore Lepay, came and talked to campus. There's a lovely talk that she gave in the ES department over in Ostrov a few years ago. There she is, talking about food as a teacher. I'm probably going to make you listen to this at some point in the semester. We're not going to do it now. But I got to realize a personal dream of mine, meeting her, and I cooked a meal for her. And all those people in the photograph there, I cooked a meal for everybody there. Vegetarian meal based exclusively on locally sourced foods and foraged foods. That was kind of fun for me. So I'm not really a starstruck person, but I was pretty excited to meet her. She's an interesting person because the thing about Frances Moore Lepay is, you can have a lot of people, academics, liberal academics up here talking about data and talking to like college students who are on track to have privileged to be here and have resources to go on and lead the lives you have. The message that she is giving and the demeanor that she has is one that is geared more towards like middle and lower income, like middle America, people that aren't necessarily empowered or privileged to be in an environment like this, and doesn't have the same kind of academic message in sort of the same way that like a faculty member may. So she was pretty interesting, and I think pretty effective. She's worked in a lot of places in the world. Interesting person to learn about, Frances Moore Lepay. Has anyone here heard of her before? A couple of you? Yeah, you? Were you there? No, you were. Yeah, you're too young. I'll give you one last quote of hers. She wrote one of her more recent books that she wrote with her daughter that was in that first picture. It's called Hope's Edge. She said, hope is not a noun. It requires, it is an action. You become hope by taking action. And I particularly like that idea, that notion, with all the things that the challenges that present themselves to my generation, your generation. It can become overwhelming sometimes, but to never lose hope, because you are hope when you overcome your own inertia. So that's a good message. It's a good thing to remember. All right. So I'm going to talk for the rest of the class about enzymes, because I wasn't going to do this now. I was going to push it off until we talk about cofactors. But I actually needed to talk about carbohydrates after I put the carbohydrate lecture together. So I got 5 slides here about enzymes. All right. So enzymes are one class of protein. Not all proteins are enzymes. We had structural proteins, filamentous proteins, etc. Enzymes are catalysts. And a catalyst is some kind of component that speeds up a reaction, a chemical reaction, in this case, a biochemical reaction, but does not get consumed by it. A catalyst is something that gets recycled. So what's great about enzymes is they're extremely specific. They're extremely specific. They only work on a very narrow, there's a very narrow band of chemistry that they do, a very narrow band of chemicals that they're going to bind to. Certain chemicals can react in perhaps a number of different ways. Enzymes allow only one output, only one of those. It takes, so for example, if at the beginning of the year your job was to create a podcast series, and I took no involvement in that, it would probably take a long time for you to get that podcast series made. And it could have been over God knows what topic related to the biochemistry of food. But I was kind of an enzyme. I sped up the process. I keep barking at you, make sure you're on a time frame. And I narrowed you down to a certain topic, superfoods. And if you came up with a topic that didn't really work well within that theme, I said, no, find another one. So I'm not making any of the podcasts, and presumably I will be just as happy and healthy a human being after class as I am before, so I'm not consumed in the process, but I'm just facilitating it and giving a specific outcome. All right? So an enzyme speeds up biological reactions. So, for example, here is carbonic anhydrase. It takes carbonic acid, It takes carbonic acid, which is the substrate. That's the molecule that's going to bind to the enzyme. You see the enzyme in the ribbon diagram above there. And it converts that substrate to 2 products, carbon dioxide, gas, and water. So here, the reactant, it says, is a biological in a biochemical reaction is known as the substrate. We see a potato. It's got carbonic acid in the potato, in the starch of the potato. You drop a solution of carbonic anhydrase on the cut potato, and it begins to bubble and foam up. That is the reaction happening, releasing carbon dioxide off the carbonic acid in the cut potato. All right? So if you cut that potato with time, the carbonic acid would hydrolyze itself into water and carbon dioxide, but it would be very, very slow. It wouldn't bubble up like that. It would be something that you would not really notice happening. But with the enzyme on there, it happens rapidly and dramatically. Okay? Make sense? Simple. All right. How do enzymes work? Well, you have an enzyme. Here are some substrates. there's an active site on the enzyme. There's the part of the enzyme, there's the part of the protein that those substrates are going to dock into, and that's where the chemistry is going to happen. So here in this cartoon, we have the substrates docking to the enzyme substrate complex, and as they bind to the enzyme by virtue of charge-charge interactions or hydrogen bonds, what have you, it orients those biomolecules in such a way as to facilitate the chemistry that's happening. All right, maybe two cysteines have to form a disulfide bridge, so it's going to orient those two SH groups, those two thiol groups, right next to one another. And then there's going to be another amino acid that's going to come in and deprotonate one of those thiol groups, and another amino acid to come in and deprotonate the second, so that they can form the disulfide bridge, for example. All right. So you go from enzyme substrate complex to enzyme product complex. That's when the chemistry has happened, and then it releases the products, and the enzyme is free to go back and do the reaction again. All right? Here you go. All right, let's look at a little bit closer. Still a cartoon. So an enzyme is highly specific. The binding pocket of the active site, the binding pocket, can only fit a very narrow range of chemical structures in the substrate. All right, so here we have the protein is chymotrypsin, and there's this hydrophobic pocket that is able to bind the tryptophan there, or the phenylalanine, pardon me. And there's a serine side chain which coordinates the carbonyl carbon in the peptide bond, holds the phenylalanine in place, and then there are some amine hydrogens that are able to coordinate the oxygen in the carbonyl of the phenylalanine. So once that phenylalanine fits into the binding pocket, it can hydrolyze that peptide bond by changing the electron density around that carbonyl group in the phenylalanine. So an enzyme can turn over one of these reactions maybe 10 per second up to thousands per second for a very, very quick enzyme. Whereas it may take days, months, possibly years for that peptide bond to break down on its own. Let's look at the energetics of this. So on the y-axis, we have energy, and along the x-axis, we just have reaction coordinate. You can think of that as time, if you want, the evolution of the system. All right. So in the beginning, we have the enzyme and the substrate, and it's at this low energy. In the dotted blue line, this is the energy it would take to make this reaction happen in the absence of the enzyme. If there were no enzyme, the dotted blue line that you see there is the energy that it would take to make the reaction happen. So the peak of that hump is what's called the activation energy. This is the energy you got to pump into the system to get that reaction to happen. And a lot of times, well, you can get that to happen by adding heat, for example. That's one way to get a reaction to speed up, is adding energy to the system. Or you put an enzyme in, and that's the purple line. So the enzyme substrate complex binds, and one of the ways you get the substrate to bind the enzyme is that binding of the two lowers the energy a little bit. It actually lowers the overall energy just a bit. The substrate's going to want to bind. Like I showed you chymotrypsin and the phenylalanine amino acid, that binding pocket that the phenylalanine is sticking that phenyl group into, that's got to be a hydrophobic pocket. And when the phenylalanine's not there, there's water in there. Water next to that greasy hydrophobic area. The protein doesn't like that. The protein doesn't like that kind of interaction. So when you stick a phenylalanine in there, you're actually lowering the energy a little bit. And that's the little dip in the road, that first dip. And then as the substrate binds, it bends that substrate in such a way that it mimics the transition state of the chemicals that are, for whatever chemical process that's happening. There's going to be some transition state. A bond is breaking, and so for the bond to break, maybe you have to bend the bond a little bit. So when the larger parts of the molecule bond together, bond to the active site, it bends the bond in the substrate, making it look more like the transition state and facilitating maybe the bond cleavage, for example. And that's how we go from transition state to the enzyme product complex, and then the products get released. All right? Does that make sense? I know this isn't super interesting, this part here, but it will have distinct relevance to food. It will, I promise. This is how we're going to digest our food. We're going to talk about digestion of food in a bit. Okay. So, the ability of an enzyme to promote this chemical catalysis is called the activity. A protein is active, an enzyme is active if it's able to be a catalyst, all right? So, an enzyme's activity is its ability to catalyze a reaction. That activity of a protein is heavily influenced by the environment that the enzyme finds itself into. So for example, most enzymes that we're going to talk about function in a very narrow pH range. So for example, here are two. There's pepsin and trypsin. Both of these enzymes are proteases, enzymes that essentially cleave peptide bonds. They're going to break a peptide bond. They're doing kind of the same chemistry, but they're going to do them in totally different milieus, totally different contexts. Pepsin is found in the stomach, and the stomach is an extremely acidic environment. Your stomach produces hydrochloric acid to help in food digestion, and in an acidic environment, the acidic amino acids like aspartic acid and glutamic acid, they are going to be protonated. They're not going to be aspartate and glutamate like they are at neutral conditions or when you're eating the food. But in the stomach, they're going to be protonated to aspartate and glutamate, for example. And that can have a direct effect on the chemistry that pepsin is going to catalyze, all right? And because of that, pepsin has evolved to basically just work in acidic environments when the substrates that it's working on are in the condition that it was designed to work on. Designed is a fraught word. But on the other hand, there's trypsin. And trypsin is a protease that's found in pancreatic juice. It works, it's produced by the pancreas, and gets secreted through the pancreatic duct into the duodenum. We'll go through the whole GI tract in a couple weeks. But trypsin is in a more alkaline environment. So we look at this chart here, and we see that the activity of pepsin is at a peak below pH 2. That's quite acidic. You guys have been drinking that kombucha at pH 3 or 4, and that seems pretty acidic, right? Well, a pH of like 1.5 is really acidic. You would be surprised at how acidic that is. That's where it works. On the other hand, trypsin works at around physiological pH. Its peak is at about 7.5. All right, so essentially neutral or a little bit alkaline. And at that pH, pepsin doesn't work any longer. So the food that you ate that was in your stomach, where's my stomach? Here it is. The food that's in your stomach, as it passes into the duodenum, the pH changes, pepsin no longer works, and the trypsin that's getting dumped in begins to work on the amino acids or the peptides that are in the duodenum. All right. Temperature also affects activity. So on the left-hand y-axis, we have activity once again. And superimposed on this, I tried to draw fraction of denatured proteins on the left-hand side. All right, normalized from 0 to 100%. And as a function of temperature. So we see in this protein here, which is some idealized protein, that there is a temperature, there is an activity peak at around 50 degrees centigrade. Body temp is about 37 degrees centigrade, followed by 2 low levels of activity. So why is activity low on the left, around 20 degrees C?
Yeah.
So that's like energy and that creates like when they start binding more because there's more energy, then like the bonds start breaking because there's like when there's less activity.
Yeah. You basically have it. Let me mix your words around a little bit. Someone else? Yeah. Any other thoughts? No? Okay. Right. So
Try to reconcile this with something else I told you today already. What else? When else have I talked about? When did I talk about temperature in these enzyme slides?
You said that like heat energy helps to like speed up the process of like biological process.
Yeah, to get over that activation energy bump. Okay. Yeah. So having more heat helps you get the energy for the activation energy, right? And that's essentially what you're saying, or at least that's the way I interpreted what you were saying. All right, that makes sense. So the protein's all folded properly at the low temperature, but the activity's low because there's not enough temperature to tap for energy to get over that activation energy hump. What about on the other side? Why is the activity low there?
up there around like 65. Yeah.
Is it like, does it require too much energy over there?
Think back to what we talked about last time.
Class. You're right, but yes, you're right, but that's like you did a bunt to first base. Let's get us home. You're close. You're on the right track. Take it, George.
Like too much denaturized protein.
You cook the proteins. Thank you. That's right. Yeah, too much of a good thing. Too much heat is going to denature the proteins, like we talked about last time in class. And you can see that there in the fraction denatured curve that I drew on top of that. At that same point, most of the protein's denatured. Most of it has been cooked. So temperature affects activity. I find it kind of interesting that at body temperature, it's not anywhere near the maximal capacity of the activity of most proteins, though. That's kind of interesting. We could speculate about that, but yeah.
Is it because, I mean, my guess is because it looks like as your temperature increases, I think about like when you're working out, your body temperature increases, and so is that like your body using, activating protein? Your body's using protein that's stored while you're working out?
Yeah, that certainly could be one. Absolutely. The one that jumped to my mind related to this. What's another thing what's another time that your core temperature goes up? When you're sick. When you have a fever. All right. Enzymatic activity can go up. Maybe facilitating reactions. I don't know. You don't want to get too close to the denaturation point of the proteins too. So you don't want to pay Peter to pay Paul. Steal from Peter to pay Paul. What's the saying? All right. I think that's it.
Okay, and right on time. Perfect. So next time we're going to start with carbohydrates, but none of that's going to be on the test. Everything up till today will be fair game. And then the podcast for this week, there's one, it's called Alpha Gal. It's pretty enjoyable. Listen to that. And if you have questions about it, you can ask me next time.
So are we also going to have a podcast quiz on, right?
The podcast quiz will be part of the whole test. There will be one question about it. Anything else? I think that's all. Oh, we do have lab on Friday, but it's going to be super short. Super short. We're going to burn the apple stuff, and we're going to flip the kefir, and we're going to see how our kombucha in the bottle tastes. Should take us less than a half hour. Thank you. Have a great day. Smile. Be nice to each other.